Hello again. So we shall be continuing with the second part of our lecture on molecular medicine. So last time we discussed restriction endonuclease, recombinant DNA, and propagation of DNA, as well as the applications of PCR. Now today we shall be continuing with the second part of our lecture, and we shall be focusing on sequencing. So there are actually two methods for sequencing. The older one is known as the chemical cleavage method. No one uses that right now. So we shall be focusing on what is known as the Sanger's method. Sometimes it is referred to as the chain termination method. Now, just like PCR, it's also based on DNA polymerase reaction. And this requires a DNA template, a primer, an enzyme, DNA polymerase, but it doesn't have to be thermostable, unlike in PCR. And just like PCR, it also requires substrates like DNTP. And of course, there's this special ingredient called the dideoxynucleotide, which acts as a chain terminator. So how is a traditional sequencing done? In its original form, it was run on four separate reactions, which means there are four separate test tubes. And each test tube or each reaction contains the requirements such as the DNA template, the primers, the enzyme DNA polymerase, and of course the substrates that will be polymerized to form the new strands, such as the DATP, DGTP, DCTP, and so on. Now, also, each reaction also contains a small amount of a particular dideoxynucleotide. So one reaction will contain a dideoxynucleotide for A, another one a chain terminator for G, another one for C, and the fourth one will of course contain a dideoxynucleotide that contains the base T. So what is the difference now between the regular substrate, DNTP, and the chain terminator? So on the left side of the screen, we have the deoxynucleotide, the regular substrate, whereas on the right side of the screen, we have the chain terminator. So let's spot the difference. A regular substrate contains a 3' hydroxyl group. Now, if you remember our lecture in DNA polymerization and replication, I did mention the significance of the 3' hydroxy. It is a requirement for the formation of a phosphodiester bond. Now, this particular 3' hydroxyl group is missing in the chain terminator called the dideoxynucleotide, which means if this particular nucleotide is incorporated into a growing polymer, we can no longer attach an incoming nucleotide to it because there is no 3' hydroxyl group, and thus it will act as a chain terminator. So how does sequencing work? So um, most of the time, a DNA polymerase uses normal nucleotide during polymerization, and the DNA molecule polymerizes or grows normally. Now, occasionally, the DNA polymerase incorporates a chain terminator, the dideoxynucleotide, which terminates further growth in that molecule. So the random insertion of the dideoxynucleotide leaves at least a few chains terminated at every occurrence. So let's illustrate this particular concept. So let's say we are in one test tube where one particular reaction is taking place. So this test tube now contains all the requirements that we need. So it has the template shown by the red arrow, several templates as a matter of fact. We also have the primers that will initiate the process of polymerization. And of course, there's an enzyme not shown in this illustration. And what will be incorporated in the growing polymer would be the DNTPs, the Gs, the As, the Ts, and the Cs. And of course, this particular reaction mixture contains a chain terminator for A, which means if this A is incorporated, polymerization stops. Whereas if any of the following regular substrates are incorporated, polymerization continues. So let's go to the polymerization process now. So if you notice, after the primer on the template, we have a G, and therefore we will be incorporating a C on the new strand, followed by a T. Now, if you notice on the template, we have a T, which means an A will be incorporated. Now, if the chain terminator is the one that is incorporated into the growing nucleotide, polymerization stops, just like in our example here. So polymerization stops, giving us now a fragment that ends with the letter A. Now, of course, this reaction contains several primers and several templates. So in another template, in the same reaction mixture, it is actually the normal A that was incorporated, which means polymerization continues until another chain terminator has been incorporated, producing now another fragment that ends with the letter A. On another template and on another primer is actually the normal A that was incorporated. So polymerization continues until we have incorporated another chain terminator. So when the process is completed, we now have DNA fragments of different length, but they all are ending with letter A. Now these DNA fragments can now be arranged from smallest to largest. How are we going to do that? Via gel electrophoresis. So this reaction mixture is run on gel electrophoresis, and then the gel is stained, producing now a banding pattern. So let's take a look at the different bands. This band right here represents the smallest fragment, if you still remember. Now we are certain that it ends with the letter A. So now we are sure that there is a letter A in our sequence. Well, there are several spaces. We don't know what the bases here stand for, but we do know that we have the next fragment, and we're certain now that it also ends with the letter A. And then there are several spaces. We don't know what they are, but we are certain that there is another fragment that ends with the letter A. So now we have the partial sequence of the DNA. So the question is, how do we now determine the full sequence of the DNA? This is where the 3 other test tubes will come in. So let's take a look at what happens when we have the 4 other test tubes. So like in our earlier example, we have the requirements, the DNA template, the one that will be sequenced, and then we have the primer that will initiate the polymerization process. So this is traditionally run on 4 separate reactions. So 4 separate test tubes containing all those requirements, including a DNA polymerase, the DNTP, plus of course the chain terminator, one for A, another one for G, another one for T, and another one for C. So let's focus on the one with the chain terminator for A, just like in our earlier example. So polymerization is taking place until a chain terminator has been incorporated, which stops the process of polymerization. So in the end, we have several different fragments now of different lengths, all of them ending with the letter A. Now, just like in our earlier example, simultaneous reactions are also taking place in other test tubes, but with different letter endings. So we shall again arrange these fragments according to size using gel electrophoresis. And then we stain now to reveal the banding pattern. So if you notice, the smallest fragment ends with the letter G. So we now know there's the letter G followed by a T, an A, another, and so on, thus revealing now the full sequence of the DNA. Now, if you wish to determine the sequence of the template strand, all you have to do is follow the base pairing rule. So nowadays, sequencing is still based on Sanger's technique, but it is a totally automated process where instead of visually taking a look at the different bands, a laser beam now scans the gel and the information is placed in a computer and the computer now prints out the result for you. So we shall be continuing with our lecture, part 3, and for the third part we shall be discussing the blotting techniques, but we shall be zeroing on southern blot with different examples. Goodbye.